I'm going to run through 3 relatively recent papers, which are all non-peer-reviewed because they are interesting, and they're all related to immune responses and the immunology of the SARS virus CoV-2. The first one is from Nature Medicine. It's a 47-year-old woman from Wuhan who traveled to Australia 11 days before presentation of her disease, and symptoms had developed 4 days before presentation. So they were able to pin down pretty accurately the time of incubation and so on. Okay, tiny bit of background on the SARS viruses. The original SARS virus, 2002, and the current SARS virus, CoV-2, bind onto their target cells using a trimeric spike protein called S1, and they bind to angiotensin converting enzyme 2, ACE2, which is expressed primarily on lung epithelium, but is also apparently expressed in the gut as well, and there's suggestions that it may produce diarrhea as well. As the angiotensin 2 converting enzyme 2 is part of the renin system, and it's interesting, I've noticed that one of the risk factors for the infection is high blood pressure. Okay, the S1 protein, the thing on the outside of the virus, consists of 4 domains, A, B, C and D, and it's associated with another protein, S2, which is involved in membrane fusion. The part of the S1 protein, third bullet point down, I don't know if you can see my pointer here, which mediates attachment to ACE2, the receptor binding domain is part of the S1B domain. And the S1 proteins of SARS, the original SARS virus, and this SARS virus are 77% identical in sequence, but interestingly only 47% homologous in the S1B domain part that is actually binding onto cells. And it has been known that neutralizing antibodies to coronaviruses usually bind to this spike protein. This has been known from the original 2002 epidemic. Right, this piece of information, which comes from a well-known textbook of immunology, suggests what you normally see in a time course of an immune response against a virus. In the early stages of viral infection, before 4 days, things like interferons and natural killer cells limit the spread of the virus, while the immune system, the adaptive immune system, has a chance to catch up. Typically, in an acute virus infection, you expect the virus to peak somewhere in the first week, and then you have the appearance of CD8 positive T cells. These are the cytotoxic T cells which kill virally infected cells, and then subsequently you get the production of antibodies which are able to prevent infection and prevent reinfection, as they may last for months or years. Typically the half-life of an antibody may be several weeks or months, depending on the type of antibody, but even after that you have memory cells like memory B cells which are capable of reactivating relatively quickly. That is how it should be. One of the ideas that came up early in this is that maybe the pathology of the coronavirus, this coronavirus, is caused by what is called a cytokine storm, where in a highly active immune response, you get cytokines released which cause hemorrhage, a lot of leakage of plasma from the blood vessels and so on, and that ultimately causes the pathology. So the suggestion was that it's actually too strong an immune response that may be particularly damaging. And that is known to happen in things like dengue hemorrhagic fever, But as we will see, I don't think that that is what is happening in this disease. Right, so going into the paper now, this is a time course of what happened with this individual. She arrived in Australia, day -7. She had onset of symptoms at day 0, presented to the hospital with an admission, and then went on to recovery, discharge, and then, sorry, I keep moving, it's a clinical follow-up. The first thing to notice is the detection of the virus from nasopharyngeal swabs, sputum, and also in the feces, which again possibly suggests that virus is being carried up on the mucociliary escalator, as you would expect, and is then being swallowed and inactivated, but there's still enough of the viral DNA present in the feces in order to be able to detect some. Notice that the assay is extremely sensitive. They've got a CT value of 45 as their limit of detection on this, which is very sensitive indeed. You've got the symptoms down at the bottom there, which is the cough and the fever, but it's interesting that the detectability of the virus has gone within about 7 days of the onset of symptoms, which means that if someone has those symptoms and it's 10 days on, you're not going to be able to determine whether they did really have this virus or didn't have this virus unless you catch a sample at the particular time when they have the most severe symptoms, the cough. And that is why the serological assay, which gives us ability to detect the antibodies for probably months afterwards, is so important. They detected antibodies in this patient on day 7, 8, 9, and 10. Looking by the ability of the serum to stain Vero cells which had been infected with the coronavirus, and basically it's an immunofluorescence assay, and you can see on the top line there that over the day 7 to day 20 you've got IgG antibodies detectable. They also say that there are IgM antibodies. Normally one would expect IgM antibodies to appear slightly earlier than IgG antibodies, and so I suspect what they are seeing here is that you do have IgM antibodies, but they're just less of them and they are less detectable in this particular assay. So on this paper they used an immunofluorescence assay to detect the antibodies. Incidentally, if anybody wants to chip in, just do turn your microphone on and cut in on this and ask questions.
People are chipping in via the comment pane, David.
Oh, right. Okay. Which I'm not seeing at the moment because I've got the whole of my screen occupied with this. I'm just wondering.
David, can I repeat my question that I typed in the box to save you going looking?
Yeah.
I was just wondering why they used Vero cells, which are kidney cells. Was that just convenient because that's their model in the lab? Because they're not terribly relevant to a respiratory complaint, I'd have thought.
Yes, absolutely. Most people are using some other cells or some kind of pseudo cells for the detection. It's merely convenience. I think they're grabbing things ahead as fast as they can. Right, I think the thing is that I'm having a little trouble. I'm not sure if I can fit up along with my presentation screen the chat box screen. So I suggest if you've got a burning question, just turn on your microphone and ask me, or someone else ask me. Right, one of the first things that they did was they looked for different populations of cells in the blood of the patient over a number of days, and they've done this by fax using a gating strategy very similar to the kind of things that we do in our own laboratory. Essentially what they've done here is they have gated a well, they started by gating for size and scatter, so they got a single cell populations. And then they separated out, they looked for B cells because they were CD19 positive, and that is on this sort of upper panel here. So this is a two-dimensional FACS scatter plot here. And then they looked for their T cells because they are CD3 positive, and that's the lower set of panels. And then they gated the lower T cells for CD4 cells, which are helper cells, CD8 cells, which are cytotoxic cells, and then they looked for various markers of subsets of T cells and activation markers. So I just put this one in here, not because you really need to know it, but just to say that effectively what they've done is a lot of gating of their cells so as to be able to look at specific populations. I've been quite selective about what I've picked out from the paper. However, this is one of the things here. So the upper graph left shows the time course of the appearance of antibody secreting cells. Those are ASCs, and they are CD19 is the B cell marker. CD27 and CD38 look for differentiated B cells, in other words, antibody secreting cells. And you can see on the open lines, open squares here, that you've got a progressive increase from day 7 going towards day 20. The T follicular helper cells are an important group of T cells because they are the ones that interact with B cells in the follicles of lymph nodes and secondary lymphoid tissues in order to promote the differentiation of the antibody secreting cells. And on the dark squared graph, you can see that they are also increasing over the course of the infection. Cytotoxic T cells, those are the CD8s, and helper T cells are also increasing, and they are looking for the subset of the activated cells because they express CD38 and HLA-DR, which is a major histocompatibility complex type 2 molecule. So they are essentially, what this is saying is that as a patient goes through the course of their infection, they have an increased level of antibody production and an increased level of active cells, as well as the T follicular helper cells.
David, is this for several patients, or are we only looking at data for one patient?
We're looking absolutely at one patient, and ordinarily a single patient would, case study would never get into Nature. But it's, and to be honest, people have looked down their nose a bit at single case studies over many years, as in what can you tell from that? They appear sometimes in, rather specialized clinical journals. But in this case, the data, I think, is so important that they've just got it out. It's not peer reviewed, and none of the things I'm presenting today are peer reviewed.
So David, do they have any, I was just going to say, do they have any data for any patients that do go into this kind of cytokine storm? Presumably they've got a totally different...
well, this patient is recovered after and therefore is presumably did not go into this cytokine storm. I think the assumption of the cytokine storm argument comes from the fact that it didn't seem to, or the severity of the disease was worse in older people and much less bad in young people. And so the idea was, perhaps they're making a strong and effective immune response, but it's actually over effective, and that's causing the hemorrhage and all the other stuff. Actually, not a great deal of hemorrhage in this, but certainly sort of plasma leakage and cytotoxicity. But it could just be, in fact, in my view, I think it is more likely that the severity in the older people is not because they've got a much stronger immune response. If anything, they might have a weaker immune response, but because they've got poorer lung function and cardiovascular function and all the other things that happen with age.
David, could I just ask, with regard to, I was surprised when I read this, how quickly the subject recovered from seeming to be extremely poorly. with regard to the timing and your expertise in this area, is that striking to you? And do you, I mean, is there anything in the study, I realize it's one person, that gives you a clue as to why it was a mild infection that they did recover from so quickly? I mean, they talk about a polymorphism, which is a risk factor.
We'll go into that at the end. It's, I mean, this person was hospitalized. Possibly they might have been hospitalized faster in Australia than they would have been in China. But I don't think we can call this a trivial, a completely trivial infection in this person, because it was severe enough for the lady to go into hospital. So it's something that they've recovered from, but it is not sort of what you might call something that is innocuous, which may be circulating in the community and in the majority of individuals.
I was just wondering if the speed of the responses here are, which is critical to getting better.
Quite in line with acute virus infections, okay. I think.
Okay.
I mean, I suspect by recovered, as in recovered enough to discharge a person from hospital, rather than recovered as in feeling completely well and ready to go back to work.
Sure.
Okay, so this from the supplementaries is looking at the incidence of a variety of chemokines, RANTES, which involves migration of T cells, MIP1 beta, which used to be called macrophage inflammatory protein 1 beta, which tends to cause the movement of macrophages. The next one is macrophage chemotactic protein 1, causes a movement of macrophages. Granzyme B is a molecule that comes from cytotoxic T cells and is involved in the external pathway of apoptosis induction when the cytotoxic T cell is killing a target cell, as is Fas ligand. And then interleukin 8 is a molecule involved in neutrophil migration. Basically, a lot of these things weren't detected, but in this particular patient, it is only one indication, and the heading up the top, no indication of cytokine storm, is mine rather than what they said in the paper, is that the actual levels are not particularly high. And the ones that you might particularly expect to see in a cytokine storm are tumor necrosis factor, gamma interferon, and interleukin 1 and interleukin 6. These are the ones that mediate the severe immune reactions that take place in cytokine storm. Now, it could, of course, be that this individual patient is not having a cytokine storm. That's the reason that she recovered and that others do. But at the moment, my view is that what we're seeing here is rather similar to what we see in any other acute virus infection, where the majority of the pathology is caused by cytotoxic damage caused by the virus rather than by the immune reaction. The final bit of this paper was concerned with something called interferon-inducible transmembrane proteins. There is a gene locus in humans on chromosome 11 which contain 5 loci of these proteins, of which these 3, 1, 2 and 3, are involved in immune defense against virus. There's another couple as well. It looks that genetically or evolutionarily the oldest one is IFITM3, and it appears to facilitate the shuttling of viruses between intercellular compartments and inhibits the fusion of the virus with the membrane of the intracellular vesicle, and therefore helps affect the release of the virus into the cytoplasm. And IFITM3 has been associated with increase of cellular resistance to RNA viruses, vesicular stomatitis virus, West Nile virus, influenza A virus, dengue, virus and the original SARS virus, as well as Ebola and Marburg viruses, but it doesn't affect DNA virus susceptibility. And previously, it had been shown that a mutation in IFITM3 was associated with hospitalization for influenza H1N1 during the 2009 epidemic. There are at least 12 identified SNPs in IFITM3, but the particular one that they found associated with hospitalization was this one called RS12252, which is present in 26% in a homozygous form of the Chinese population. So if you go down to the last bullet point there, in East Asia, the allele frequency of this is 0.5 to 8, which gives you roughly 26% homozygotes in the Chinese population. I've got this material from different sources, so I'm just putting the numbers together myself. Whereas that variant is much, much rarer in the European population, only 0.041 as the prevalence. That particular variant has a 21 amino acid truncation at the N-terminus, which is created by change in the splicing of the gene. And basically what they have noted from this is because this gene is associated with increased risk of hospitalization, because you're not controlling the release of the virus into the cytoplasm so effectively, that it is interesting, therefore, that this patient has this particular variant, even though it is moderately common in the Chinese population. So I don't think that they can say anything more about that, but certainly it's an area that people are looking into to see whether perhaps one of the risk genes for severity of the disease is one of the variants that is known to give increased risk to the original SARS or one of these other RNA viruses. I'm just finishing on that case study now. If anyone wants to chip in on comments on that, and then I'll go on to the serological assay.
If I may, David, do you know what intervention, what treatments they gave this poor lady when she entered the hospital until she left?
I would imagine that it was breathing support. I could look it up. I didn't look at the details of it, and to be honest, I'm not quite sure it was there. At the moment, they're trying a whole series of different treatments, one of which is chloroquine, the idea being, I think, to change pH of the intracellular vacuole so as to reduce the escape of virus from vesicles into the cytoplasm. There's been suggestions to try some of the other antivirals, but most of the other antivirals are things like, rather specific for influenza, where you're trying to inhibit the neuraminidase, in other words, viral release, or HIV, where you're trying to inhibit the reverse transcriptase. Well, that's clearly not going to have any good effect in this. So at the moment, I think the only ones that they're really looking at are things that may be able to interfere with virus release from the vesicles into the cytoplasm. And certainly I don't think they tried anything on this lady. They just followed her and gave her support.
Okay, thanks, Dave. It's just something I came across in the past when there was some clinical intervention of early fluids had shown to prevent cytokine storm when it's done early, in particular at pre-hospital. I was just curious as to whether they did that and what colloids they used. But yeah, thanks for that, mate.
Yeah, okay.
David, and is it Sala? It does actually say that she had, her management was intravenous fluid rehydration. It's at the bottom of the first column.
Oh, thank you. Yeah, well, that makes a good indicator how they managed to avoid a cytokine storm. Okay, thank you.
Okay. I'll move on to the, where are we? The serological assay. Well, this was sort of good news, came out at the end of last week on I think it was Friday. And basically, it's an absolutely standard ELISA in which you have the target protein of the virus, the S protein, which is what binds onto the receptor on the lung epithelium. And they got 4 coronavirus 2 patients and quite a sizable panel of controls. Interestingly, one of their control serum is from a convalescent patient for another coronavirus, which binds onto the same receptor on the cells. And basically, they use 2 different types of antigen, either the whole spike protein, that's on panels G and H, or just the receptor binding domain of the spike protein 1b domain there. Essentially, what they're finding on all of these is that they've got a perfectly good and specific assay for it, effectively using an ELISA where you've got logarithmic scales on these graphs, and what they're doing is they're measuring effectively area under the curve, which is a slightly more sophisticated way of looking at titer of the virus. I mentioned to Mark that with our new ELISA virtual assay, we could probably reproduce this almost instantly, which is as a teaching tool, which would be quite a nice thing to do. Okay, so I don't think there's anything much more to say about that, except that the antibodies that are developing in these patients are binding, I think, as we do expect, to the receptor binding domain of the, or some of the antibodies, to the receptor binding domain and to the spike protein. And they're quite clearly distinguishable from control, so it should be an effective way of detecting seroconversion.
David, could I just ask on that? It's Hilary. Does it mean anything that you get better binding with the whole spike protein than with the subunit?
Yes, it probably does mean something, in that the spike protein has got a better configuration, and therefore I suspect it's the affinity of the binding that is slightly more effective there, so that effectively the dissociation constant is a lower value, which means effectively that the area underneath that curve comes to be larger. So there is some debate in the paper as to which of the particular antigens they're using is the best, and I think they come to the conclusion that maybe the M spike is best because it's got the best differential between the controls and the SARS coronavirus patients. But to be honest, they're all working pretty well. Yeah, I mean, it looks like the M spike is better than the I spike, and it looks like, as you say, the whole protein is better than the just the receptor binding domain.
Thanks.
Yeah, sorry, go on. Someone else came on. Okay.
Oh, yeah, sorry. Can I ask you, David, it's Francesco. Is there any evidence in the literature of how long the antibodies last after seroconversion? And I suspect there's been no study on asymptomatic patients, right? To see how many of them.
There can't be any at the moment. I didn't show the data on the different antibody subtypes from that patient, but it is in the supplementary material, I think. Essentially, you would expect, if nothing else happens, an IgG1 antibody would have a half-life of several months. So after an infection, you would expect, even as the antibody declines over a period of months, up to a year, to still have significant and detectable levels of antibody in the serum. So my guess is that with nothing else happening, just the antibody naturally decaying, that you would have protection for, my guess would be about a year, but that's just my thought on it. Other subclasses of antibodies tend to decay rather more quickly, but even if the antibody itself decays, you still should have B memory cells, and you've got activated T cells which can reactivate much faster, on the assumption, and actually we're going to come on to talk about exactly what part of the spike protein these antibodies might be against in a minute, on the assumption that the virus itself does not mutate, so that the spike protein no longer binds the antibodies.
David, this is not one. I think it's a related question to Frank. I have read in the press, I think it was The Guardian, so I haven't had time to check it myself, that the immune response to coronaviruses, that typically it wanes after a while. Is this what, so it's not as kind of like as a strong an immune response as...
Nacho, can you come a little closer to your microphone?
Can you hear me now? Okay.
That's better. Yeah.
Yeah. Sorry. No, I was saying that I have read in the papers, but I happen to have time to check it, that the immune response to coronaviruses are typically not very strong, and the immune response wanes quite quickly. Is that correct, or I haven't had time to check it on any biomedical books or anything?
There are a variety of coronaviruses, and we know that viruses do vary in, things like norovirus, the immune response, but that's a gut-associated virus, tends to wane rather quickly, but that may be rather different from a virus that primarily infects the lung, where I would expect the response to be more persistent.
Okay, thanks.
Hi, this is Samir speaking. So I had a quick comment in regard to that. I know that for the common cold at least, there's other coronaviruses which show a seasonal element, and that would suggest that there'd be some sort of waning immunity to these types of viruses. Certainly when you look at flu as well, there is a seasonal element to that, and I would doubt that the immunity would last, because these viruses part of the reason that there is such a variety, that you get repeated cold viruses, is that there is such a variety of viruses, so that you're not protected as the virus changes or as a new virus arises by the antibodies that you've got from the previous one. The seasonal effect is seen both in the northern and the southern hemisphere, and is probably more related to the rate of transmission in cold weather rather than the some specific thing about immunity waning related to the season, in my view. Should we go on to talk about the monoclonal antibody that they've produced? I think if you got around to this paper, and I've been reading quite quickly on these things, so you may well have picked up other things in the paper that I haven't, is just what an extraordinary amount of work has been done in a relatively small amount of time. So this particular monoclonal antibody, which is in a paper described on Monday, what they'd done was they had taken mice which have the human heavy and light chains of the antibodies coupled to a rat FC region. These are referred to as H2L2 mice, and they were immunized with the S protein, the external domain, from
different strains of coronavirus, because what they were interested to do was to potentially to see whether they could induce cross-reactive antibodies. Now, if you will recall, the receptor binding domain of the S protein is in the S1b domain, and so what they particularly interested in was antibodies. They got out 51 monoclonal antibodies here. So some of these were binding to the S1a domain, so those they thought, 23 of them, were less useful. The ones that they thought were most useful were the ones that were binding to the S1 protein but not binding to the S1 domain. And out of the group that they got there, the group of 22, they picked this antibody 47D11. It binds to the spike protein, the external domain, but it did not bind to the S1a domain. And then what they did was they took the cDNA encoding the variable regions of that, and then they cloned that into expression plasmids containing wholly human IgG1 heavy and kappa light chain, that is a kappa chain region. So effectively taking what was a hybrid antibody, half human and half rat, and making it into a wholly human antibody. I mean, I find it just staggering that this can be done in such a short amount of time. They then transfected the DNA for the fully human now, heavy and light chains into HEC293 cells, and they produce some monoclonal antibodies. The first thing is, this is a binding curve looking, yes, it's an ELISA, looking at the binding of this antibody at different concentrations, and you can see it's still detectable at around about 1 nanogram per mil. Binding to the two top lines where it is binding are to the B domain, which is the one they want, whereas the binding to the A domain is almost non-existent. Now, the most important thing on this is they looked for the ability of the antibody at different concentrations, so you've got a concentration curve along the bottom, to interfere with the infectivity of cells. There were viro cells again, infected either with the original SARS virus, SARS-CoV, or the current one that we're concerned about, SARS-CoV-2. And you can see the greyish line is an isotype, IgG1 isotype control, which is having no effect on the infectivity, or a marginal effect on the infectivity on the covariant coronavirus 2. And you can see that there's an inhibition, 50% inhibition somewhere between 1 microgram per mil and 100 nanograms per mil in both cases, about a microgram per mil for the SARS, the current SARS coronavirus. Now, this is interesting because while everybody is working flat out to develop an active immunization, and that's the conventional way we would do for any immunization, passive immunization, which is the treatment of people with antibodies, has been done for many years and is still used for occasionally acute things such as dealing with snake bite. In other words, you've got to neutralize the venom very quickly. It is theoretically possible, therefore, maybe to use a passive immunization with a humanized antibody in severely ill patients to limit the infection over a short period. Now, one would not ideally do this because you would normally go for an active immunization with a protein or with an attenuated virus or something like that. And most of the vaccines that they're trying to develop at the moment have put the DNA for the spike protein into an innocuous virus. However, the one advantage that this might have is that the pharmaceutical industry is already geared up for the production of monoclonal antibodies. It has the facilities. They're used for treating cancers and various other things. And so it might just be advantageous in the short term before an active immunization comes into use, something like this. Now, the interesting thing about it is, although it is inhibiting the infection with coronavirus, it's not doing it in the conventional way. This graph here is showing the ability of different antibodies to inhibit the interaction of the S protein with the target antigen, ACE2. And in this, they've taken two monoclonal antibodies, 35F4 and 43C6, which are known to inhibit infection of the original coronavirus because they interfere with the interaction of the receptor binding domain and the ACE2. And you can see on this graph here that the 35F4 and the 43C6, these two descending lines in the middle of the graph here, as they inhibit the interaction between the protein and its target, they're doing it quite effectively at around about 10 to 50 micrograms per mil. The interesting thing is that the 47D11, which is on the blue line, although it is inhibiting infectivity, is not affecting the interaction of the S protein with its target. And again, so this was, actually I'm going to shift through that one because I've slightly forgotten what was happening on that particular graph. But let's just go on to the very last thing on that. So they said that the antibody neutralizes the viral infection, but it's not due to inhibition of the binding of the S protein to its receptor. In other words, a novel mechanism. Now, they said in the discussion to the paper that they're surprised that this antibody was cross-reactive against two different SARS viruses. Certainly it had been, the immunization schedule used two different SARS viruses, so would potentially promote cross-reactive antibodies. But nevertheless, because this part of the virus varies a great deal, you might not expect to find cross-reactive antibodies. And so what they're suggesting is that this antibody they've produced binds to another part of the S1B domain, a core part of the S1B domain. And the suggestion then is that it could be disrupting the overall structure of the spike protein, so that in some way that it can't trigger the release. Now, if you remember, the S protein has two components to it. It has the S1 protein, which is doing the binding to the receptor. It's also got the S2 component, which is involved in the fusion of the membrane so that the virus escapes. Now, it could be that if you interfere with the interaction between the S1 protein and the S2 protein, that although you don't prevent the virus being taken up, you do prevent it actually getting from the vesicle through into the cytoplasm. So I think that's where we are with this particular chunk of stuff, and I think that's about as far as I've been able to read, but there'll probably be a number of other papers coming on very soon. So I think what I'm going to do is I might just sort of progress back to this rather hopeful stuff here and just open it up for any general discussion.
David, this is not that we were kind of like on the diagram. So we were talking about whether it would be possible to have.
Nacho, can you come closer to your mic again?
Sorry. In the chat box next to the screen, we were discussing whether passive immunity approaches could be used in a pandemic situation, and Samir was saying that, and Francesco was saying, sorry, pharmaceutical companies can scale up, and Francesco was saying that maybe the limiting step is the clinical trials with new antibodies.
I mean, I suspect the clinical trials will be shortcut. I might stop well, actually, I'm going to leave. I'm going to stop presenting temporarily so I can see what's going on in the chat box. I hope. We'll see. I think the thing is, it's just the sheer quantities of it. You know, for the small number of people who are relatively small number of people with a you know, a particular tumor or a particular autoimmune disease, producing autoantibodies to perhaps neutralize cytokines or target a tumor is manageable. Passive immunization of this sort is not going to be usable on a large scale. Apart from anything else, you have to infuse the antibodies intravenously. The amount that is actually being required here is around about something like 10 micrograms per. You might need several milligrams into a patient to get a decent amount, a workable amount into them to actually do anything. This particular antibody is an IgG1. It's not secreted. Ideal if you're trying to protect the lung, you would need an IgA antibody, which might go across on the secreted, on the, you know, the secretory systems for IgA. So I don't think it's going to be a feasible, except perhaps in hospital situations where you might have somebody, and this might just tip the balance, but it's not going to be a generally usable thing. I mean, if one just considers, you know, what the cost of an active vaccine might be, which might be, you know, a pound or two per dose, whereas the cost of this, which would be hundreds or thousands of pounds per dose, it can't be used on a wide scale.
David, slightly naive question. I'm just thinking about people who are asymptomatic. In your experience, if people are asymptomatic, how does their immune response compare to somebody who was very ill? Is it equivalent?
I don't know. I mean, the thought, and especially for an unknown virus, I really couldn't say. I mean, the trouble with asymptomatic people is that they don't actually go to a doctor to have their symptoms checked or taken, or, you know, or anything like that. So I think it's not just this virus, but the information on that is very sketchy for everything.
Yeah, I'm just thinking about the concept of herd immunity going forward, and whether people can actually be infected several times. Which has been speculated, depending on, of course, you know, the immunity that they build up.
Well,
so
I wouldn't. I mean, a lot of people have been concerned about that. I think this is just my view, so this is, you know, please don't take this as gospel. But I suspect that we will have a considerable amount of immunity for if you catch it, for this virus for quite a long while. However, if the virus mutates significantly, then it would be possible to get another variant of it. A bit like flu, really. Yeah. However, there's usually some residual immunity for influenza related to the fact that even though you don't produce exactly the right kind of antibodies, you've still got T helper cells and cytotoxic T cells, a pool of them, which are able to control virally infected cells. So you're probably not quite as badly off as when you are having to deal with a virus that you and the whole human population has never encountered before.
I actually saw one paper where they tracked the viral load in China in something like 60 people, and the people who were asymptomatic were the ones that were transmitting for the longest, for up to 3 weeks. I'll try to find the paper again and can send the link around.
So would that suggest then their immune response, they hadn't mounted a very profound immune response because they hadn't cleared the virus from their system?
It could be, or it could be that the immune response is the virus hasn't gone into the lungs. I mean, it always, if you get this virus in your gut or on your tonsils, which is, a good solid part of the immune system, it isn't in what you would call the target organ here. So you might have a sort of low-level infection, I suppose, have a reasonable immune response, but you are possibly not isolated. I don't know the answer to this. I'm just guessing. Katja, what do you want to do about, are we going to have a journal club next week?
I think Fra has got a speaker for next week. I'll check whether that's all going ahead. If not, we should have a journal club. So we tried with Fra speaker to have it, to have him present via Skype. They're going to test it beforehand, whether it all works with the equipment. Otherwise, we should continue with journal clubs if we can't find an external speaker.
Well, thanks very much for setting this up. It shows what can be done with the technology. I think it's really excellent, and it's also a slight morale boost to be in touch with everybody and